Hi all, this is my review on Chapter 4 for Bio 101. Remember that the complete notes for my notes, my personal notes, this is a walkthrough, is going to be in the question thread for week 1 for these notes. Okay, so Chapter 4. Basically this is going to talk about cells. So first we know that in the 1600s the first microscopes were used. By 1665 Robert Hooke describes the cell in oak tree bark. 17 to 1800s, we discover cells are just pretty much everywhere. Mid-1800s, cell theory is described. So it's a nice, long history of cell theory. So what is cell theory? Cell theory is that all living things are composed of cells and that all cells come from other cells. You know, I love for you to look up germ theory because it's kind of a spin on this, but we're going to focus on our cell theory. Remember, some things are weird like viruses and other potentially things that might be alive or not alive, but we're going to focus on cell theory. So if you got cells, they showed up from other cells. You know, and then don't forget that Large Hadron Collider where we're looking for the God particle. Looks like we found it, right? Okay, so now prokaryotic cells. These are cells that are characterized as organisms that are bacteria and archaea. So basically little one-celled critters running around. Eukaryotic cells are things that are like protists, plants, fungus, and animals. So in general, I think of prokaryotic as a little more simplistic. They're going to have smaller DNA, if they have DNA, than our eukaryotic cells, which is what we're made of. And there's a couple videos. I have a little checkpoint in the chapter, which I'm going to ignore. I gave you a little chart here to help explain this. So here's what's the same between the prokaryotic and the eukaryotic and what's different. So the same as we both have that plasmid membrane. That's what encloses our cell. We're both DNA. We both have ribosomes. Okay, now what's different? As you can see, prokaryotic is hecka older. They're smaller. That's why a lot of the gene studies you hear about tend to be in the prokaryotes. There's just less to have to deal with. And they have a nucleoid rather than a nucleus. Nucleoid is not separated by a membrane. Nucleus is it has that separate membrane. In the book, and most of what stuff we will be discussing will be more on the eukaryotic side. So we will have those lovely membranes. Great little video. Oops, too far. Here we go. Plasma membrane. So that is the thin membrane that goes around the cell and it regulates what goes in and what goes out. Ribosomes are tiny little structures that build proteins. Remember from the previous chapter that proteins do like everything in our body, so we need them. Organelles, you're going to have a DQ on this. Those are membranes, enclosed structures that perform specific functions. So we call them little organs. So they're like the organs of your body, they're the organs of the cell, okay? And it lists some various organelles, ER, Golgi, vacuoles, chloroplast, mitochondria, anything with a membrane. Nucleus is, we care about, remember that's eukaryotic, and that's what's holding our DNA, so it keeps that really important information separate. Cytoplasm is the entire region of the cell between the nucleus and the plasma membrane. So it's basically just all the lovely stuff that just other than the nucleus, just floating around, having a good old time. Then there's another little checkpoint. I'm just flicking way too hard today. Okay, then let's talk about plant versus animal. So these are all going to be eukaryotic, but now plant cells are slightly different than animal cells. Plant cells have the chloroplast, that's why they're green. They have that cell wall because they don't have a structure. They have a central vacuole. Animal cell has an extracellular matrix. It has lysosomes, which occasionally can be in plants, but pretty rare. Centrioles, and it has the flagellum. Those are different things. Phospholipids, we care about these because they're going to be in the membrane, right? So phospholipids have a water-loving and a water-fearing side. The reason this is important is they can point around so that the right sides are in the right area. We have a phospholipid bilayer, which is right below here. Is a really good picture. So here you see that The phobic ends stick together, while the philic ends, remember that means water-loving, are on the outside. We also stick in all sorts of little things in our membrane, and this will ease with different substances traveling through the membrane of the cell, right? Stuff got to go from the outside to the inside, to the inside to the outside. But we want the right amount of movement, or we will either starve or bloat the cell. So very important to eat that right amount. And having those stuff stuck in there, like icebergs, is part of that mosaic model. Okay, extracellular matrix. This is around cellular animal cells, and it's like a sticky, secreted coat, and it holds together cells together in tissues. It's protective, supportive functions. So instead of having, you know, our cell walls hitting each other, we've got this little matrix. Cell junctions, these are structures that connect to other cells, and they're in animal cells. So whatever needs to hold us together. The nuclear envelope is the double membrane bordering the nucleus. Chromatin is what are long DNA molecules that are associated by proteins and form fibers. Chromosome is one chromatin fiber, right? So all of them together, chromatin. We're talking about one specific one. That's chromosome, and this is where our genes are. The nucleolus is the prominent structure within the nucleus. Is a site where the components of the ribosomes are made. So most of the stuff we talk about where ribosomes are made, that's going to be in our nucleolus. And ribosomes, again, protein synthesis. Remember, proteins do just about everything. Let's scroll down. DNA programs protein production in cytoplasm by transferring its coded information to a molecule called an RNA, messenger RNA. So we got a little M in there because it's a messenger, and that tells you it's moving around, right? Communicating like a messenger. Like a middle manager, the RNA molecule then carries the order to build this type of protein from the nucleus to the cytoplasm. So inside the nucleus to outside. So still in the cell, but not in the nucleus. The mRNA exits through the pores in the nucleus envelope and travels to the cytoplasm, where it then binds to the ribosomes. As a ribosome moves along the mRNA, the genetic message is translated into protein with a specific amino acid sequence. Okay, that's all for the notes part 1. Let's go to part 2. Here is an image of what we just talked about, right? So the DNA goes to the mRNA. The mRNA plunks through the nucleus into the cytoplasm, and there the mRNA hooks up with the ribosome to create our protein. So some organelles are connected to each other either directly by their membranes or transfer of membrane segments between them. This is a lot with our endomembrane system, especially the ER, endoplasmic reticulum, I can't talk today, endoplasmic reticulum, or the Golgi apparatus, lysosomes, and the vacuoles. So we're just going to call it the ER because really, I'm just going to say it wrong. So it is one of the main manufacturing facilities within the cell. It produces an enormous variety of molecules. So if you want to know where something's made, it's probably in the ER. So rather than saving people at the emergency room, here we're making stuff. There are two compounds that make up the ER. That's the rough and smooth. One's rough, one's smooth. It is nice when they actually call things what they are. So the rough ER refers to the appearance of this organelle in electron micrographs. The roughness is due to the ribosomes that stud the outside. Okay? And these ribosomes that are the rough part produce membrane proteins and secretory proteins. So they're secreting or their membrane, right? Some products manufactured by the rough ER are dispatched to other locations in the cell by means of transport vesicles. So they're going to have to move around. Membranous spheres that bud off the rough ER. Okay, so let's talk about the smooth ER. Smooth ER doesn't have the ribosomes. Is a diversity of enzymes built into the smooth ER membrane enable this organelle to perform many functions. One is to the synthesis of lipids. So basically rough, think ribosomes, like it starts with rough, right? Rough, ribosomes all start with R, they make a bunch of proteins. Smooth, you're going to have to think of just the other option, which is fats or lipids. Now the Golgi apparatus is a refinery, warehouse, and shipping center. So what that means is it works with the ER. The Golgi receives, refines, stores, and distributes. So basically ER makes the sucker, then the Golgi takes it and makes it even better and sends it out. Products that are made in the ER reach the Golgi in transport vesicles. One side of a Golgi stack serves. I feel like you guys can go through this in more depth if you choose this for your specific DQ response, right? Generally, it's just going to fix stuff up and send it out. The lysosome is going to be, I think of it as the stomach. It's where the digestive enzymes are. And they also can be called food vacuoles. Depends on what you're doing. So what are vacuoles in general? Vacuoles are sacs that bud off from the ER, Golgi, plasma membrane, and they have a bunch of variety of functions. One is lysosome, sometimes it's just to hold stuff. It says in certain freshwater protists, there's contractile vacuoles that pump out the extra water. A central vacuole in a plant is a versatile compartment. It stores organic nutrients such as proteins stockpiled in the vacuole's seed cells. It contributes to the plant's growth by absorbing water and causes cells to expand. I really think generally of vacuoles versus lysosomes. I mean, they all kind of go together, but lysosomes specifically digest stuff. Vacuoles to me are kind of like that room of requirement, isn't that what's called in Harry Potter? Like you need a room to stick something, that's your vacuole. Then we're going to get into photosynthesis a little bit. So plants have chloroplasts, and this is where our photosynthetic cells of plants and algae are, so they perform photosynthesis. So basically chloroplasts turn light into energy for the plant to use. Mitochondria, these are one of my favorites. They're separate from the rest of the stuff in your cell, in your body. They're going to have their own kind of DNA, which makes them really unique. But in terms of what they do within the cell, they harvest energy from sugars and other foods, molecules, and convert it to another chemical form of energy called ATP. ATP is what we put our energy into, and we break it apart, put it back together. Okay? The cytoskeleton contains lots of fibers and proteins. One fiber that we care about is the microtubules. Microtubules are straight hollow tubes, and they're composed of proteins. See, proteins never ends. Flagella, these do a whip-like motion. Think of them as the tails on sperm. That really gives you the mental image. Cilia are like little hairs. I think of them as, you know, rowers, a bunch of oars on like a Viking boat. And they move stuff. Generally, they'll be lining, like your windpipe, it says over here, and they'll push the food in the right direction, or the mucus, whatever they need to do. I'll let you read all that. Okay. And then let's just do the quick compare and contrast cilia and flagella. Cilia and flagella are the same basic structure and help move cells or move fluid over cells, right? I think most of the stuff we talk about, I always think of the flagella as moving and the cilia as moving stuff over, but it can be either way. Cilia are short and numerous and move back and forth. Flagella are longer and often occurring singly, okay? Hopefully this helped you understand the week 4, or chapter 4 reading, week 1, and it's all about cells and what happens inside of cells. Try not to focus too much on specifics, but get the general concept. What does all this mean? How does this relate? Can I think about what's happening in the cell and think about how that might transfer to what's happening in the actual human body or the animal body?